Playa Venado in Veracruz, Panama. Perfect location, teeming with Crepidula species we use in lab. We chose this rocky zone as our field collecting site. In a moment, we will see a kit used to identify 19 different digestive enzymes. In preparation for this experiment, the species collected were brought to our marine laboratory here on Knox Island. This is my third internship in Panama City working with the Smithsonian Tropical Research Institute and the Collin Lab, and is ultimately a very good learning experience for me.
Some species analyzed include Crepidula marginalis, very common in Panama. Planktotrophic development, where larvae hatch and swim in the plankton for an extended period. Crepidula atrasolia, common in Florida. Direct development with large eggs having individual crawl-away hatchlings. Crepidula onyx, direct development with nurse eggs, which are consumed by developing embryos, also common in Panama. Crepidula ustulatelina, unique lecithotrophic development with non-feeding larvae that have a brief planktonic period but settle quickly. Crepidula incurva and Bostrocapulus calyptiformis. Both have planktotrophic development and are found in Panama. With these species, we will compare the activities of digestive enzymes within developing embryos and veligers at 5 different stages during development. We will identify any large shifts in enzyme activity throughout development and also compare the changes across the distinctive development strategies. The developing embryos, which are incubated around the propodium, must be collected from the adult snail. The best way to do so is to use a forceps or pipette to pull them from the mother. The embryos are collected and stored in a negative 80 degrees Celsius freezer. Let me tell you about my strips. The API-ZYM strips are a semi-quantitative assay to determine the existence and activity of 19 digestive enzymes. To begin, we homogenize a small sample of embryos, or hatchlings, in order to create a uniform liquid. The protein content is measured, and the sample is diluted and used in the strip. Each strip is incubated for 4.5 hours at 73 degrees Celsius, where special substrates react with the enzymes in the solution. After the incubation, 2 reagents are added to stop the reaction, which results in the color we use to analyze enzyme existence and activity. Each well has a specific color, and the intensity of the color indicates the activity of the enzyme. Here are the spectrophotometer results for well 6. It has a very high peak, which indicates high activity levels for that specific enzyme. After we measure each well, we plot the value for each stage from egg to veliger. This particular enzyme, alkaline phosphatase, clearly increases throughout development from the egg to the veliger stage. In our research, we then compare how the enzymes change for each species and also how they compare to the other closely related species with different development strategies.